Rain. Not so much fun.
Hello, everybody. Welcome to Q&A with A&V. Welcome back. I'm Vincent Racaniello, and with me from New York, Amy Rosenfeld.
Hello, how are you today?
Well, you know how I am because we spoke this morning, right?
Well, we spoke multiple times. I was being polite. We spoke
I'm
multiple
well.
times, right?
Since I saw you, I've been well. How are you? You have a good day?
No, not so good.
Not so good.
Not so good.
I got a
It's
haircut.
fine.
I just came back from my haircut.
That's good. I didn't even make it to the gym. I'm not so happy.
I'm sorry. You were
It's
supposed to
okay.
run this afternoon.
I know, but I had a lot of work to do.
Okay.
It happens. Oh, I have to turn that off.
Someone
Yeah.
asked if Lori Garrett could do a This Week in Public Health.
Yeah,
However,
I
I
think
told them
once a month.
I ran it past you and you thought it was a good idea, right?
Once a month. She could cut once a month or once every 6 weeks. Sure.
So I will ask her, but I have a feeling she has her own thing she said they're renting space in Dumbo, right? What does Dumbo
Yeah.
stand for?
Downtown under the Brooklyn, between the Brooklyn Manhattan Bridge. It's a very hip area in Brooklyn. It's very
And she
expensive.
said they're going to do some podcasts, but maybe she would like to join us. We'll see.
Well,
And
just because she does one podcast, you do like 11. No, it's not me playing the piano. You do 11 podcasts, so it's a different audience.
Apologies for
it's a
the
different.
piano. It's my daughter upstairs who plays whenever she wants. And normally she doesn't play more than 2 minutes, but tonight she's playing longer. I texted her to stop, but she's not looking at her phone while she's playing the piano. I hope you enjoy it.
It's fine. But I think that there are important topics to discuss with Lori, like
Yeah, I agree.
how to make it such that the public health infrastructure is completely independent of the whim of the executive. So she talked about it being here in the city at the whim of de Blasio, and we saw it at the whim of the former president, right?
Yeah.
Which was not good.
No.
And then there are other things to talk about,
Yeah, I think I will approach her in some time. Amy, what was your title for the Times piece? Tell them your title, Amy.
Oh, it was variants, scariants, and human behaviors of concern.
They didn't like that. I mean, we didn't even pick the current title, right?
No, we didn't pick either one of them.
In fact, let me pull up the article because I don't even
What
remember.
got edited out? So a lot got edited out to fit a non-science audience.
Yeah. Here. Human behavior during the pandemic is more important than any COVID variant. That was not our title.
No.
But that's what they went with. They told us they experiment with different titles with their social media team to see what works. Not what the authors of the piece want, but what works. Which seems, we complain a lot about headlines, and this is why, because the authors typically don't make them. I think this is a flaw in the journalism world, frankly. Yes, Amy.
I mean, I guess they're supposed to know what is going to draw people's eyes and get them interested to open up the article. I guess that's what their skill is.
I mean, variance, scariance, and human behavior of concern is perfect.
It is, but what we get drawn in and is attractive to us may not be attractive to my aunt in Florida.
Okay, fine.
I don't know. I mean, I'm not a publicist, and I don't really. You have to spend a lot of time and going to groups and taking surveys, and that's just not what I do. I'm not interested.
Well, there was, as Amy said, there's a lot of science taken out, and maybe I'm going to look at we signed an 18-page agreement with the Times, but neither of us read the whole thing, so I want to
Who
see
has
if
time to read 18 pages?
of legalese, right? So
Exactly.
if possible, I can republish it for those of you who haven't seen it. But maybe I could republish an earlier version, Amy, that has the science in it. That would be good, right?
Yeah, we can work on that.
Okay. I really enjoyed Lori's TWIV. It was nice to hear how she came to be concerned about many things that we never notice. Yes, it was a good episode. Did you like the Garrett episode, Amy?
Yeah, but I think it's just I think there were missed opportunities to actually delve into like topics that instead of covering so many different things, just cover fewer topics more in depth. As I said, like I think it would be interesting to hear her perspective on how we should reorganize the CDC and the NIH from being, at the whim of an executive. I think it's interesting that she brought up the polio vaccine and was really adamant and kind of mad at the fact that the Gates Foundation refuses to give any money into surveying. But if she knows anything about polio here, then she knows that the CDC refuses to survey here, even though we've been declared eradicated. And we've talked to them about that multiple times, remember?
Yes, for sure.
And so the reality of it is it's probably here, right? You would agree that it's most likely here. We just don't want to find it.
Oh, yes, polio is
Um,
in the U.S. for sure. We just don't want to find it because we wouldn't know what to tell people, right?
right. Um, I think I think that uh, another thing is like for the next pandemic, an idea of how to prepare. So I agree with her about the diagnostics. We don't have good infrastructure. We didn't really have a good diagnostic. But also, should it really be about the social and stuff and how to give a more coherent picture in a polarized situation? Because we're probably not going to become less polarized, right?
Is it possible to get vaccinated in New York City,
Yes,
Amy?
you can just walk in, and actually, they've extended the days. So there's a vaccine center at Penn Station. There's one at the Vanderbilt building, which is across the street from Grand Central. They were all supposed to close the midpoint of this month, and now they've all been extended to like July 8th or something. So you can just walk in and say you need a vaccine and roll up your sleeve.
Welcome back, Frank. Thanks. Frank is one of our mods. I believe Vanity Nutrition is also here. Thank you so much for keeping the discussion civil. It's great.
Good.
Steph says, I love when she said in the pandemic role playing, they never envisioned being taken down by world leaders. Touche.
Well, yeah, I don't think anybody did.
Nope. Hello, Vanity Nutrition. Missed you
Yeah.
last week. Thanks for coming back.
Yeah.
Oh, yeah, this story about the Chinese highways through the jungles. Yeah, that was very cool. I didn't know that. Very
Yeah,
cool.
I did.
Yeah, I think
They wanted to be in like I think it leads almost to like a sea that connects it to the Indian Ocean. So they wanted connections not just to the Pacific and the South China Sea, but to the other side, and that made it a lot that allowed them to do it. And I think that that's part of the reason why we're looking in the wrong spot.
I hope this week in public health could happen. Yeah, I mean, I think public health is an important one, and if Lori doesn't want to do it, we'll find someone else, I think.
Well, I'm sure you can probably ask Brian's wife, or Emily, who's in the Colorado Public Health
But
Department.
she doesn't have the world view that Lori
She
has,
might.
right?
You don't know. I don't know. I haven't talked to her. Never met her. She might have a very large worldview. She's not a dummy.
I love this comment. The best part of the week is about to begin, and the
It's
most
cute.
informative live stream of the week. Thank you, folks. It's just very nice that you like what we do.
It's great.
Lovely, lovely. Okie dokie. Lori killed it last week. Okie dokie. Oh, do we get closer to understanding the outbreak in Brazil when seroprevalence was claimed to be very high? Remember that, Amy?
Yeah, I vaguely remember it. I'm not sure that they were telling the truth about the seroprevalence being as high as it was, but I remember it.
So someone wrote into TWIV subsequently from Brazil who said the problem was it was a blood bank survey and that selects for a certain population. It's not a random selection. People who give blood are not random, so it's not a reflection of the total population. That may be one of several problems. I don't know.
Well, it sounds like it.
Yeah. Is there a non-paywall link? I don't believe so, but if you want the article, you can email me and I'll send it to you. And yeah, that's no problem. I have a PDF, which I just showed you, actually. Right? This is the PDF, which I made in a nice format. Let's see, at the end, they actually cited Amy being a 22-decade virologist and me with TWIV. TWIV shows up in the Times. How cool. Now, that wasn't in the print version, right, Amy?
No, it's in the print version.
Oh, yeah? Very good. Okay, H. Zoo. Is it true that certain viral infections during pregnancy is associated with birth defects?
Yes.
Absolutely. Can you name one, Amy?
Zika.
And I can name rubella virus and cytomegalovirus.
Yep.
So what's the mechanism of Zika virus, Amy? You used to work on that, right?
Yes, I did. So what do you mean? Which part is the the mechanism of what?
How does the virus cause congenital defects?
Oh, it causes it impairs brain development. So it can kill the radioglial cells, which are required to generate not only the size of the brain, but the neurons of the 6 layers of the brain, which then facilitate um, gyrations and other stuff. So some of the pathogenesis is, it enhances calcium deposits, but really it's the death of the radioglial cells that line the ventricle.
And Amy actually published the best paper on that
I
a number of years ago.
Yeah,
Beautiful
I published
paper.
a good paper on it. Yeah, so it's
and,
interesting.
And the mechanisms differ according to the virus. I'm not sure we understand for CMV and rubella virus, but basically, in some cases, the virus gets in and reproduces in cells, as Amy said, for the Zika radioglial cells. But for other viruses, it's probably different cells. But yes, they do cause. And SARS-CoV-2, I don't think there's any evidence that it's a teratogen, right, Amy?
Oh, no, there's no evidence.
Okay, how is it possible that antibodies pass from mother to baby through milk? Wouldn't proteases in saliva, stomach, intestines destroy them?
Well, no, I don't think so. I think like milk is full of fats and stuff.
Yeah, they are passed.
So I think they are passed, but I think that they're kind of like protected because they're in such a fatty solution.
Yeah. They're very effective, and many studies have been done showing that breastfeeding reduces the incidence, say, of rotavirus infections in infants before rotavirus vaccine.
Yeah. I mean, that's part of the idea about how polio was, right?
Yeah.
And definitely part of the idea about our timing of 68. So, yeah.
Have you read any good papers on the Delta variant?
Well, when a good paper comes out, we'll read it.
Yeah. No, so far, there's some preprints, but we're waiting. There's nothing published, peer-reviewed yet.
I'm not even sure that just because it's peer-reviewed, it's good.
Yeah, that's true. It's better. It could be. Yeah, you're absolutely right. But it's what we're required to do, right?
I'm not required to do anything. Okay. Yeah, we could go with that.
ACE2 versus ACE, they work in opposite directions to control various pressure-sensitive systems, so I'd expect the distribution to be similar. Are there ACE-specific viruses? Well, okay, I'm not aware of any that bind ACE. Are you, Amy?
No.
Yeah. Whoa, here we go. What are the properties of adenoviruses that makes them the choice for several vectored vaccines? One of them is that, that you can put rather large genes in them. You can actually take out all of the adenovirus genes and put a lot in, but for the spike-based vaccines, you don't need to do that. And they're easy to manipulate, and they're easy to grow in large quantities and purify. Those are all good properties for a vector. You can also make them so they don't cause disease in people. All that good, Amy?
Yeah, but I mean, in theory, you could do a lot of this for like other viruses in theory, but you can't really because like you can't really put too much, you can't really take too much out of polio, right, to make it a vector because
No,
otherwise
no.
it's not processed and assembled properly.
Yeah, you can't take
So
very much out, yeah.
there's stuff like that is really that makes adenovirus very amenable because it really requires very little to assemble the particle.
Do mammalian cells have a CRISPR-like system to maintain genomic integrity? I think Amy can do this one.
Not
Right?
that I'm aware of.
No, they don't, but they have other systems to maintain genomic integrity, right? They
Yeah,
have proteins that detect damaged DNA, for example, right?
yeah, but so do bacteria. I mean, much of like what Rod works on, the Rod 52 and all this stuff, they're orthologs of like Rec A, B, and C, and Rev A, B, and C, and Mut S, Mut L, Mut H. So they all have a lot of the same properties.
Yeah.
Um, but we also have, we're also the nucleus segregates the chromosomes away, and various other, we're coded in different kinds of proteins, various other things.
By the way, one of Rod's people is taking my course.
Which one?
I don't know. It's a Japanese name.
Oh, really?
And
I know that Megan wanted to do it, but she didn't have time. She
yeah.
signed up for Borkman's class instead or something. She's going to take your online course that's live in the fall.
I'm teaching online. I'm teaching a virology summer course to Columbia students this summer. Just started this week. And I will do an online virology course this fall from the incubator.
Yes, that's why we have
I was
it.
thinking maybe I should set up an area where I can stand up and give the lectures like
You want a podium?
I don't need a podium. I just would like to stand up because I think it's good to stand up when you're sitting working at the computer.
So you want a standing desk?
I might want one in one part of the studio and a regular desk in another. Can I get both?
Yeah.
Okay, last week we talked about the 4 different dengue types. Are these variants or strains or none of the above? Dengue, very common here in Brazil. They're serotypes, right, Amy?
Yes. They're not variants and they're not strains. They're serotypes, which means that they elicit different types of antibodies. They have different epitopes.
Aw, thank you, Joe.
That's very
kind.
nice.
Very kind. All right. Reports stated genomically identical Delta variant infected people merely passing by both indoors and outdoors. I don't believe it for a second. It's anecdotal, mass media nonsense. Just nonsense. Yes, herpes simplex also does.
Yeah, they're torch agents. It stands for toxoplasma. Others, which is herpes and which is HIV, rubella, cytomegalovirus, herpes, and then Z is Zika. So they're torch agents.
Right. My bangs. So when I got the haircut tonight, the lady was combing it to the side. I said, "No, you've been cutting my hair for 20 years. Don't you realize I wear it in little bangs so I look younger? This is the Pete Townsend look." Oh, Jeffrey, hello. Mrs. Jeffrey's going to be our fundraiser.
Well, that's good because now hopefully, he can get it together and start fundraising
Yes,
better than your
we have to talk.
publicist. He's not
Jeffrey,
going to like working with me because the minute he doesn't do something is the minute I'm going to yell at him.
Jeffrey, we have to
It's
talk.
true. It's true. That's
Maybe
the way it
sometime
works.
tomorrow. Maybe sometime tomorrow. Tomorrow afternoon, maybe around 2ish. I'll be in touch. I'll email you. Thank you, Jeff.
You'll email him. Is that like when I say to you, did you email Peter to publicize tomorrow's TWIV, to publicize the things? And you say, yeah. And I guarantee you it still hasn't been done.
I don't know what to do when I say I have to email someone. What do I do? Do I write it on a sticky pad and then I forget the sticky pad somewhere? Look I wrote here 42 centimeter bottle height. That's the height of the microwave I have to get for the lab. Oh boy. It doesn't work on a sticky. I don't know how else to remind myself, so I just keep it in my head. And
maybe you should make it an alarm.
Yeah, the alarm will go off. I'll shut it off. And
well, you use the alarm every day to get up.
I don't know what to do. Anyway, all right. I'm going to email Jeffrey. What's your opinion on Pfizer first, Moderna second? Beautiful.
Fine.
It's all good. Absolutely. Yeah, the piano stopped now. Sorry. Yeah, she's. It's not bad. It was low, right?
Yeah, it's fine.
I wonder if you have any thoughts about Pfizer dosage levels announced for about 12-year-olds having adult dose. What do you think of that?
They seem to tolerate it. They seem to tolerate it. It seems to be fine.
Yeah, I think it's okay. Yeah, with vaccines, we tend to do that, except we increase doses for older people like me, right?
Well, for certain vaccines, you get high-dose flu.
Yeah.
You don't get high-dose tetanus.
No, not all vaccines, okay. Absolutely. Thank you, Jeffrey, for your contribution to the incubator. The incubator. Oh, Amy and I talked to a woman this morning, a scientist at Mass General.
Harvard,
who's from Australia, and she said, yes, they're making a mess of the vaccine program. They didn't get enough vaccine. Yeah, it's a mess. Yep. I wonder if you listened to the CDC ACIP meeting about myocarditis and mRNA vaccines and what your thoughts are. I did not. Did you, Amy?
When was the meeting?
I don't know, but...
well, if my question is when is the meeting, then obviously, yeah.
You know, most likely Daniel...
Obviously, I didn't listen.
Most likely
I'm sure
Daniel will,
does. I'm sure tomorrow you can ask Daniel for a synopsis.
So here we go. Statement following CDC ACIP. Well, okay, I don't want to read it now, but I will ask Daniel tomorrow to summarize it. I'm sorry, Simon. Let's see. This is a credulopathic remedy. Love that name. Caveat about reading efficacy data. J&J claims efficacy of 67% against symptomatic, but a third of cases are asymptomatic, so doing nothing also has efficacy against symptomatic COVID of 67%. I don't understand that. I don't understand it.
I don't, yeah, I'm not clear.
Thank you. The Times piece was fantastic. We put a lot of time into it, didn't we, Amy?
Yeah, took us like 2.5 weeks.
Amy she says we have to do this today, and we sit down in my office typically and do it.
Yeah, tomorrow we're doing the grant.
The Delta variant has a couple of my family members worried. You shouldn't worry about it. It prevents the vaccines. Even J&J prevents moderate to severe COVID, at least in the South African variant. Have you seen any studies, J&J versus Delta, Amy?
I don't remember that one particularly. I know that they've looked at the AstraZeneca, they've looked at Pfizer and Moderna. They're all fine. I don't see why J&J would be any different.
Yeah, Jane. So I would not worry. Tell them that the real key is it prevents moderate to severe disease death. So that's what we said in our op-ed, essentially, that get vaccinated will prevent you against any variant. And you're okay.
Well, so like who Dan Baruch today had a paper. Theodora has had a paper. They all describe how when you get vaccinated, part of the immune response automatically degenerates into protection against the variants. It's not a problem.
I recall you saying there's a difference in transmissibility in virology versus epidemiology. However, I am a human etch-a-sketch. That's funny. I don't know what
So
it
this
means.
is where a certain individual that you talked to today screwed it up.
Yeah, in epidemiology, they say this virus is moving through a population quickly. They call that transmission. In virology, transmission is the spread of a virus from one person to another. And if you want to say that a virus is biologically more transmissible, you have to do the right experiments. So what they're seeing epidemiologically is a more fit variant spreading through the population. And let's say there's an outbreak. Well, the outbreak happens now because people are getting back together, they're not masking, and many of them are not vaccinated. So whatever virus is around is going to infect you.
Right. And the other thing they started off with it's more transmissible, all this stuff, and it's like a founder effect.
Yeah.
They don't understand founder effect either.
Founder effect means the virus is in the right place at the right time. You start an
Basically.
outbreak, and whatever virus is there is in the outbreak.
Right.
Anything behind the variant being transmitted by fleeting exposure? No. I know we answered this already, but I want to answer it again. There's nothing behind it because this is the kind of stuff the press.
Seconds rather than minutes? Seconds?
Yeah, it's ridiculous.
Really?
It's ridiculous. Thank you, Zachary, for your contribution. Seeing many criticisms of CDC saying vaccinated people don't have to mask. Zero COVID people calling vaccinated who don't mask immoral.
What are zero COVID people? Who are those people?
There
Never
are
heard that term.
zero COVID people. I don't know what that means.
Yeah, I've never heard that group. I've never heard that term referred to as a group of people. But yes, she has come out and reiterated that you do not have to mask if you are vaccinated.
I think it's fine. And if you are worried, I mean, I wear masks to certain places. It depends on the local transmission levels, right? If the numbers, if infections are high, you should be careful.
Right. But like here in the city, it's really low. It's like,
but at Columbia, we have to mask indoors, right, Amy?
you have to mask certain spots, not and stuff. When you're like on the elevator, you have to mask, yes.
And in our lab, we have to mask, right?
No, not really. You don't have to. But
yeah. But you wear your mask all the time.
Yeah. We go out in and out, right?
Yeah. What's your favorite wine, Amy?
Well, I like very expensive wine, so I would like a Barolo. But I like a Malbec and a Cab. I also like wines with I like bubbles, so I like champagne or prosecco.
The headline, I guess it's the name of our article, approximates what I tell people when they express worry about vaccinated. Get vaccinated. Okay, yeah. If Delta gloms onto ACE2 better, why would it be more transmissible? Yeah, I agree. It wouldn't be necessarily. Glomming onto a receptor better is not going to necessarily make a difference. But people who don't understand that say, oh, it's glomming on better, it must be more transmissible. That's the narrative, right, Amy?
Yeah, but I don't know that they use the word transmissible for that, or they use the word infectious.
More infectious, yeah.
Yeah. So I kind of think of infection, it's a little extreme, but I kind of think of it as you're transformed. Once you're transformed, you're transformed. You can't be more transformed. That's ridiculous.
Luc Montagnier asserted that variants are a consequence of natural selection due to vaccinations. And water has memory. Yeah, right. Montagnier, I'm sorry. He's wrong. You know, unfortunately, he has a Nobel Prize, so people think he's right.
Yeah, but so does a certain other individual who has a Nobel Prize and is screwing it up also. So just be quiet.
Here, this other person maybe says it, what we don't want to say.
Yes, that's one way to say it, but there are other ways to say it. And there are other people who fall into that category who are colleagues, right? With Nobel Prizes.
Thank you, K8, for your contribution to the incubator. Most polio is mild. Most infections are mild, yes. Only 1% are paralytic. Right, Amy?
Yes.
Oh, you can have a drink. I said, right, Amy. Everybody take a drink. Or what, if your favorite beverage, water, coffee, anything else. Amy, Vincent, these pieces are important since the Times has a broader reach than any journal. The disconnect between science and lay is what resulted in the distrust misinformation. I hope so, Jaimito Frog. I hope so. That's why we wrote it. We wanted to reach people.
Yeah, I mean, I think though the distrust and misinformation is maybe not necessarily a disconnect by science and lay people. It because I think scientists have a role in that. Um, and yeah, so I think that scientists have to take some responsibility.
Agreed. Is Novavax available anywhere? Not that I am aware of. Hasn't been authorized yet. Do
Oh, is that a
polio
question?
No, it's just a statement.
Oh,
Well, is Novavax available? That's a question. Yeah, I said no.
uh,
I didn't think it was available anywhere, right, Amy?
right. It's not. I didn't know if there was a question.
Yeah.
Sorry.
Do polio vaccines last for life? Do they still give it to children? They do give it to children. Here in the U.S., we use the inactivated vaccine. Does it last for life?
It's hard to know.
Hard to know. It lasts a long time. It lasts many years. And I've seen articles from Mark Palanche, Amy. He says, in some countries, OPV immunity wanes after a certain
Yeah,
period of time.
I know.
But a boost with IPV sometimes can bring it back.
No, I'm aware of what Mark says. And Mark is really smart, and he's one of the good guys at the CDC.
We have an independent Federal Reserve. Why can't there be an independent CDC NIH? Challenging is the current political...
Actually, we don't really have an independent Federal Reserve. The head of the, or maybe it's isn't the Federal Reserve part of the Treasury? And the head and the Treasury is the Secretary of Treasury is appointed by the president. Also, the people, what is Yellen head of, or she was on the board. There are like 7 banks in the country that make policy that come together to define interest rates. What's that called? Vincent?
I don't know. Federal
Well,
Reserve?
well, if that's the committee that like gets together to discuss like whether or not you should raise interest rates and stuff, that is definitely not independent. That can be subjected to that can be subjected to presidential pressure.
I thought, I was going to ask Lori about that, but she seemed to, I don't know, I didn't in the end because I thought her answer was it's impossible, it can't happen. But it's a good question.
But that's the question that we just discussed.
Yeah,
when she said, that de Blasio fired all of the public health people and put MDs in their offices, and now the New York City public health department is a disaster.
Yeah.
That mimics exactly what Trump did, right? So that was the time to discuss whether or not there was how to make it independent so that it wouldn't be at the whim of the executive. Although, if you put it under the legislative branch, I guess it's at the whim of people like McConnell and Rand from Kentucky who don't seem to believe that there's any need.
I read asymptomatic COVID infection could still show evidence of organ damage if a fully vaxxed person becomes a breakthrough case. Does the vaccine protect against that? Well, I don't know about this asymptomatic and organ damage. Do you, Amy?
I guess that's like long-term COVID. Most long-term COVID
Yeah.
patients were asymptomatic.
Yeah. So a fully vaxxed person, no, most vaccinated people are going to be infected at some point, but they may not have symptoms. We don't know if it's going to protect against these long-term at all. We don't. It's been too soon, right, Amy?
Right.
Thank you, Mike. I won't make a comment. I will move on. New preprint investigates AZ and J&J clot issue, states evidence is due to incorrect administration of vaccine. Should go BIM. Some cases goes into the vein or artery. Daniel talked about this.
Yeah, there was a lot of controversy about pulling back the plunger and stuff. So let's all not. You can just listen to Daniel, and we don't really need to rehash it because it was a little uncomfortable.
Daniel went into this on one of his twivs some time ago. Yeah.
Two of his twivs.
Could you explain in a way I can pass on why it's better to wait longer between the initial vaccination and the booster? I ask because I recall Amy saying the longer the better. Do you want to explain it, Amy?
I can try. So when you get vaccinated, it takes a certain length of time for you to go through the whole process, to have the T cells engaged, then for the B cells to be activated, to go back to the lymph node, make antibodies, come out, do some selection so that you only have the high affinity ones that survive, and then to go and make memory B cells. And so that's not an instantaneous process. That's like 6 months of work in your body, right?
Yeah.
And so a booster is supposed to reinforce the development of memory B cells. So my logic is if you give it too fast, you're not really reinforcing the memory B cells. So
the timing of the first and second doses were come about just to do the whole vaccine development quicker for this pandemic.
Right.
3-4 weeks, but normally you would wait longer.
Right. That's what I've always said.
Yeah. Yep. Absolutely. Please invite Nels to TWIV. Yeah, he's been on TWIV before. We talked about his picornavirus of zebrafish. Amy, have you seen his OAS paper? Well, anyway, I thought he might want to come and do that one. It's quite interesting.
I haven't seen it, or maybe I have seen it. I forgot. I know that there was recently a big OAS paper. I don't know if it's him.
Do you see normalcy after this winter with less mask wearing and less thinking about the pandemic from the everyday person?
Depends on what country you're talking about.
Totally. How about the U.S.?
Oh, we'll forget about the pandemic by the time Halloween occurs, because young kids will be vaccinated, and then you'll hit the 70% needed for herd immunity of the population, right?
Yeah, I think there will be winter outbreaks though for years to come.
But there might be winter outbreaks because there's always going to be the people who are too young to be immunized, right? And they potentially could get infected, like kids under 5. Yeah. And then there's always going to be a population that's immune compromised.
Sure.
Blah, blah, blah. So you're always going to it's going to be like flu, right? You're going to have like 100,000 cases a year, but you're not going to have you're not going to have a 6-month period where 600,000 people die again.
No. And with influenza.
Well, at least not here in the United States.
People have accepted the flu season, right, with the disease and death and it's routine. People don't talk about it, and that's what will happen to COVID in the U.S. But it will take longer in other countries, as Amy said. Adam loved our piece. Everything explained clearly and understandably without being too dumbed down, but the cynic in me makes me doubt it will change many minds. You might be right. You might be right. No substitute for Lori. I followed her for 20 years. Well, if she says no, we have to have a substitute, right? I mean, Lori is a writer with a very good oversight of issues. Not many writers have that oversight.
Yeah, but she's not really a writer. She's really a biologist.
Well, she started to train, right, in science. That's correct. And then she went to writing, but yeah, she understands things pretty well. My friends are 70 and vaccinated, going indoors to bars and dancing. I'm afraid to participate. I'm 78. Am I being too careful? What do you think, Amy?
I don't know if I would have said I'm being too careful. I think you should do what you're most comfortable with. If you're feeling left out, I think it's fine to go. Everybody is vaccinated, and that's the point of being vaccinated, is you get to go back to doing the things that you were doing beforehand.
Yeah.
If you don't, if you're nervous, then you're not going to enjoy it. So then there's no point.
I haven't gone dancing at a bar in a while.
When was the last time you went dancing in a bar? When you were 20?
Yeah, probably. But look, I would not go to a crowded place right now, even though I'm fully vaccinated. I just want to help to reduce circulation. So, but as Amy said, if you feel uncomfortable, it's fine. You're not being too careful. I heard they can't make a respiratory syncytial virus vaccine because of enhanced disease. Now, the first vaccine they tried in the 60s caused enhanced disease, so that put a damper on RS virus vaccine development for many years.
They actually developed one. It's in clinical trials.
Yeah, so now there's one in clinical trials. And we did a TWIV in Nova Scotia a couple of years ago with a lady who is working on that. And
I'm not.
Excuse me, let me finish. And she is advocating the idea of immunizing mothers, and then they would pass the antibodies on to the children, because you have to get the kids very early on, because this is a virus that can strike in the first weeks of life. So that would be one approach. What are you going to say?
Yeah, but that's not the vaccine. That's not the vaccine that's being tried. The vaccine that's being tried is the pre-fusion stabilized.
Yes, I know. I didn't say it was being tried. It was just another approach. Yeah, this
another approach of what?
I don't know. Another RS virus vaccine in development. The lady from Johns Hopkins was working on it.
Yeah, but whatever.
Thank you very much for your support, H2. That's very kind of you. Very kind of you.
In order to say that there's another approach, you have to tell them the first approach.
No, I don't have to tell them that.
Well, then it doesn't make any sense if there's another approach.
What's the explanation for waves of infections? Simply human behavior? It's a combination of that and environmental conditions, immunity levels. It's not one simple explanation for the waves of infection. Okay. Ooh, did you read about the case of Jamestown Canyon virus being reported in New Jersey? I did not. Did you read that, Amy?
No.
It's an encephalitis virus. It's a bunya virus spread by mosquitoes. Let's see. 8 days ago, yeah. Sussex County, a Sussex County resident in his 60s tested positive for JCV after onset of fever and neurological symptoms in May. First detection in this state, and only this year, and only the second ever reported in New Jersey. It was also one in the same area in 2015. So cool. Well, not cool, but yeah, it's interesting. But it's only one case, which is good. Been fully vaxxed for almost 3 months. With this Delta variant still going around, do I need to mask inside and continue to avoid eating? As we said, if you're fully vaccinated, you can do as you wish if you feel that you want to be more careful, but you're going to be protected against moderate to severe disease caused by any variant, as the New York Times opinion said. There's been a lot of talk on sequence variants in the UK. Are these consensus? Is there a good study on intra-host variation in the quasi-species compared to between hosts? No, but it's normal to see variation everywhere. And we're doing a lot of sequencing, so we're going to pick this up. Do you know of any good studies on these, Amy?
Good studies of what?
Intra-host variation in the quasi-species compared to between hosts. So looking at
There
one
was
host.
Wait, I'm discussing. There was a paper that did look at the development of variants within a person, compared, and then most of them discuss about whether or not it comes from somebody else.
Did you hear that a third dose of Oxford AZ vaccine efficiency goes up a significant amount? Yes, I did hear that. Did you? You sent me that paper, I believe, or preprint, Amy, right?
Yeah, we did. It's 8:45. 2 more. Then I have to go take my cells out of trypsin.
Has it been proven that severe outcomes from COVID in healthy people are caused by high exposure to virus?
There is no correlation between the amount of inoculum and severity of disease.
Are there any self-replicating, replication-competent viral vectored vaccines in general or for COVID? Well, there are replication-competent adenovirus vectors, and I believe at least one COVID vaccine is based on that.
I thought that was disproven. I thought that was the cause of a lot of uproar about why Brazil and other places should not use the Johnson & Johnson or AstraZeneca one.
Yeah, that was the Sputnik one, yeah.
Yeah. And then it was like, well, they didn't really know how to interpret the plaque assay or something or other, wasn't that, or something like that?
Mm-hmm. Yeah, that's exactly right. Like less than 50 PFU, the detection limit or something like that.
Yeah, and then Condit had some discussion about why he never does an undiluted thing on plaque assays, which I didn't really follow.
Some viruses, when you make cell extracts, they're toxic to the cells. So our viruses are not, so that's not an issue.
Yeah.
All right, here's the last one. Why are you so sure the lab leak was inconceivable when there's still no evidence of a middle host?
Why do I need an evidence of a middle host?
Well, I guess his logic or her logic would be if there's no middle host, then it had to leak out of a lab. But I don't think that makes any sense.
I don't understand why that is the logic, because you go so if they listen to Lori, she tells you about how these people walk around barefoot and various other things, right?
Yeah.
And if you go into the caves or into the mine shafts to take the guano and make it into fertilizer, right, you're aerosolizing stuff. And it's like dander, right? That's how allergies to dander occurs, is that you aerosolize stuff, right? And many of these viruses have now been shown to be able to bind to ACE2 directly from the bat, right?
Yep.
But the paper that you, Alan, and I did, and then Lympha's paper, right?
Yes.
So why do you need a middle host?
Don't really. Not at all. I mean, because for SARS-1, we had it, so people assume that it has to be for this. But, yeah, it doesn't need to be a middle host.
But for SARS-1, it was in one, we can say it was pieces of it were in one cave. But you can see from the paper that you, Alan, and I did that pieces of it are not even in the same species of bats, right?
Yeah,
There
that's
was
right.
something that was an RP, right? Isn't RP
Rhinolophus.
some other bat
Yeah,
species?
yeah, Rhinolophus P versus Rhinolophus A and others. Yeah, absolutely.
Right. And one of them was 100, the ORF1B protein was 100% identical to SARS-CoV-2,
Yeah.
right? It was from like RP. And then there was the RN, right? And the RN had like the core 6 amino acids in spike or something. Remember that? Like it was like 403 or something,
Mm-hmm. Correct.
right?
Yeah.
So you can imagine if those bats share some kind of common habitat and they, let's say they fight each other and bite each other because they want the space, you can imagine that the viruses go in between the animals, right?
Mm-hmm.
Animals, that's what animals do, right?
Oh yeah, big time.
And then it would recombine in, bat number 3. Oh, this is going to be fun.
What's the matter? Is it raining?
Yeah, and I forgot my umbrella. And now it's lightning. It'll be fine. I'm
All
going
right.
home
All
after I
right.
finish splitting my salts.
All
All right,
right.
tomorrow we need to work on the grant.
Okay. Thank you.
All right.
Good night.
Bye.
Bye
Good
bye.
night. Bye.
Okay. Listen to the TWIV that's dropping tomorrow about spillovers and so forth. There's a lot of information about that. Anyway, back to Naivri's comment. So inconceivable is a hard word, right? I don't see any evidence for a lab leak. That's why I say it's highly unlikely. There's plenty of evidence for natural origin, and as we see more and more SARS-CoV-2s in bats, it gets more and more likely. And there are many other reasons why a natural origin is the most likely event. Okay, let's see. There was another one. Here we go. You said you didn't believe the variants are more transmissible. So let me just say, belief, it doesn't really have a place in science. If I say believe, then I'm misusing it. I would say there's no evidence that the variants are more transmissible. Epidemiologists see outbreaks and they're fueled by variants. And so they say, they must be more transmissible. And they actually calculate a transmission index. But that equation, as I've said before, fails to, it includes human variables, and they fail to update it with human data. So, the human interactions are driving the outbreaks. It's not the variant. So what do I define as transmission? Person-to-person spread of the virus, which is governed by biological properties. But in an outbreak, of course, if you're in a room with 20 people, as opposed to in a room with no one, that's going to determine transmission, right? And that's the part that's not being considered. How would you demonstrate more transmissible? So what I'm thinking is the variants are more fit. Fitness can be caused by a lot of different things like antibody evasion. And a small increase in fitness could drive a variant to outpace another variant. How would you demonstrate it? This is really hard. It takes years. Let me just say that for no human outbreak of a virus, no one has ever demonstrated an increase in transmission over the course of the outbreak. Very difficult to do, always circumstantial evidence. Let me bring up the Ebola virus outbreak in West Africa in 2015. Early in the outbreak, a variant emerged with a single amino acid change in the spike protein, and eventually that virus infected everyone in the whole outbreak. Many people said it was more transmissible, but it was never proven. In the laboratory, that virus grew better in cells. But what does that mean? What kind of cells? Here, for SARS-CoV-2, you'd have to at least use human respiratory cells in the lab using infectious virus. Okay, BSL-3 experiment. Nobody's done that. Animal transmission, ferrets, hamsters. You can't use that many animals. The results would be inconclusive for people. So it's really hard to prove transmission. You could prove higher fitness in the lab. You could examine why a virus is more fit. But connecting it to transmission is really hard. So that's why I object to people saying they're more transmissible, more infectious, because there's no biological evidence for it. You have to have evidence, so don't say it. What you can say is they're more fit and they're moving through a population. Okay. Okay. Will you, if you do online course, will you issue certificates? Well, I have no power to issue anything of value. I could make a certificate, saying you've completed the microbe TV virology course, but it's not going to do you any good except make you feel good. So I'm not a degree granting institution, right? I'm sorry. For reviewer selection, when is someone's experience sufficient to review versus a conflict of interest? Well, the way we do conflicts of interest now in review, either of grants or papers, so anyone at the same institution is out, right? Because that's a conflict of interest. If you've worked with someone in the past 5 years and published, that's a conflict of interest. That's out. However, if you do work in the same field, that's not necessarily a conflict of interest. And you're probably the best person to review someone else's work in the same field, right? Because you think about it all the time. Unfortunately, you're probably also their competitor, and you might take advantage of the review system to delay their publication, to delay their getting a grant. Really nasty stuff which happens, and it's a product of the way we fund science. It's competitive. There's not enough money for everyone to go around, and it's very competitive. So people do things that aren't so nice. And this has been extensively studied, unfortunately. But anyway, those are the rules for conflicts of interest. So, yes, stand, sit, desk. So, for example, now I'm sitting down. I've been sitting here all bloody day. I got up to walk around every now and then. I walk my dogs, blah, blah, blah. But I would like to stand up now. And my desk is fixed. And my camera's fixed. So I'd have to have and in the studio, I'm going to build something where I could stand. I'm going to have 2 stations, one where I stand and one where I sit for doing different things. That's the beauty of the incubator. I can do what I want. Will COVID end by eradication or turning into an endemic? Oh, endemic for sure. No eradication of SARS-CoV-2. It's around to stay because it's in animals most likely. It's in people, and we can't immunize everyone. So, yeah, it's here to stay. We're going to have outbreaks every year. We have 4 endemic common cold coronaviruses. This is probably going to be another one of those in a certain number of years. Yeah, it'll be a mild virus for sure. We should get certificates for fans who I followed Twitter for a year and a half. Sure, I could do that. No problem. I could design a nice certificate and send it to you. You bet. Oh, ivermectin. Look, listen to Daniel Griffin on ivermectin. My opinion is, if you want to take it, go ahead, because it's cheap and it's very safe. It's a really safe human drug. By the way, my wife worked on ivermectin at Merck for 25 years, so I know something about it. What is needed is a good randomly controlled clinical trial of about 4,000 people, and I believe that's going to be done, because there have been a number of studies, some of which show efficacy, some of which don't, but they're too small to be conclusive, but they're big enough to say we should do a big clinical trial. So I think it should be done. I totally agree. But, that's expensive. A 4,000 person clinical trial is many millions of dollars. But I understand it's going to be done through a number of people I've talked to. So that's great. That's great. Threshold for herd immunity. So there's a formula, which I don't remember right now. But basically, it's based on the transmission index, the R0. That is how many people can an infected person infect on average. It's 2 to 3. And, Laurie Garrett said Delta's 7. I don't believe that at all. That calculation is wrong. It's 2 to 3, which means you need from 50 to 70% of the population immune for herd immunity. So I would say 70% is a safe number. And we can approach that for sure. As usual, the media is continuing to confuse me on Delta, wondering, am I still fully protected if I'm vaccinated with J&J? So, the word fully protected is a little vague, right? What do you want to be protected against? Against getting infected? No. Nobody's going to be protected against getting infected a year after vaccination. Are you going to be protected against getting the sniffles? Probably not. Are you going to be protected against moderate to severe disease to death? Yeah. Yes, you're going to be protected. So don't worry about it. Don't worry about it. Don't listen to the media. They don't know. They don't get it. They have no way of parsing all the information that's coming out there. And they always talk to the wrong people, like Luc Montagnier, who doesn't know what he's talking about. Email yourself instead of sticky notes. Yeah, you know what I've done? I've sent myself texts. But as soon as I send them, they show up and the dot next to them disappears. An email, well, Jeffrey, my future fundraiser, he emails me, and then I get so many emails a day that in the morning the email may be at the top, and by the afternoon it's at the bottom, and I'm never going to see it unless I scroll down. I really could use an assistant to go through it all and make lists for me, and I don't. Oh well. I'm still talking about the piano. Man, I must be so far behind. My email is Vincent@microbe.tv. Yeah, it's a public email. You can do that, and you can ask me questions. You can send money, whatever. You can't do it through email, but you can email me and ask me for my address if you want to send a check. Thank you, Ditching the Grind 3, for your contribution to the incubator. Yeah, we will publish some form of the unedited article, okay, in the coming weeks. I think that would be informative, yes. Do you think that children who are not yet eligible for vaccination should be wearing masks inside? It depends on the infection rate wherever the kids are. If it's low, which it is in many parts of the country, then and if they're surrounded by vaccinated people, then you don't have to do that. But if infection rates are high and they're going to be in a room with unvaccinated people, then they should, yes, to be safe. How's it going on proving that those who have been double jabbed can spread virus? I don't think that we know yet. Those studies are ongoing, but my suspicion is that they will show that. I mean, that's what herd immunity is about, that vaccination inhibits transmission, right? So I think if you're vaccinated, you'll be infected. You may get a mild disease, but the amount of virus you're going to produce and shed is going to be much lower, so you won't be transmitting. And so therefore, herd immunity will work. And then that elicits a thumbs down because I say herd immunity will work. It's my opinion. You don't have to take issue with me. Are the different symptoms reported for Delta due to increased replication, vaccination, or media invention? So there's slightly different symptoms because the people who are being infected are younger. In general, now, people who are infected are younger than they were a while ago, and they have different symptoms. That's why it has nothing to do with the variant. And this, by the way, is what Daniel Griffin said last week on TWIV. Getting the jab, it doesn't make you bulletproof, but you're not going to die. You probably won't get severe disease, so that's good. Thank you, Parallax, for your contribution. Case counts in Scotland are skyrocketing. My parents are 68 and 70, pre-existing conditions, but feel being fully vaxxed, they are safe. They're protected against moderate to severe disease, yes. So if this is an acceptable thing to do, that they can go to a crowded place without a mask, yes. As I said, some people don't feel comfortable doing that, so it's up to them. But technically, they are safe doing that. If they have pre-existing conditions, though, depending on the nature of them, I'd be really careful for sure. For sure. Zero COVID just not happening in my view. I mean, you could have it as a goal, but hey, look, we've been trying to eradicate polio for 40 years. 1980 it began, and we're almost there, but it's still, pottering on for sure. It's my impression that epidemiologists only come up with hypotheses. Yeah, they do. They use models to say, looks like this is happening. Yes, you're absolutely right. And that's why you can't prove transmission increase, so you have to do the experiments with the virus. And the press doesn't get that. They just don't get it. And that's the point of our article. But in Europe, zero COVID refers to the position that the only acceptable approach is to drive cases to zero. I think that's a bit extreme because you're going to disrupt people. There's got to be a balance between allowing people to have normal lives, right, and cases. It doesn't have to be zero. That's too much. Some people argue that eradication is not really a realistic goal because it imposes, it's a huge cost and resources, right? The polio eradication campaign has been extremely expensive. So it may not be the best strategy. Just reduce the number of cases. Okay, so Marion, what would be the right experiments to show that a variant is more biologically transmissible? So, as I mentioned this earlier, so I don't want to repeat it, laboratory experiments, but they can never completely confirm it because people are different, right? Replication in cells, animal models to look at transmission and disease. So you'd have to do observational studies in people. I mean, ideally in people, you'd compare people infected with a variant and the ancestor, but you can't do that. Most of the time, the variant displaces the ancestor. It was possible early in the outbreak, but no one did it. It was really an opportunity that's passed, unfortunately. Marianne Koopman's on TV. Her role is a mix between science and vaccine champion. On the variant, she was correct. We work with the assumption of 1 times more contagious. So it's an assumption, but it's not proven. And there's no need to work with that assumption. Just get vaccinated, and you don't have to work with that. I don't understand the logic at all. So I don't agree with her. Am I right in thinking asymptomatic spread itself is something that adds to the fitness of a virus as it helps to add to the long chain of unreported cases? Now, it doesn't add to fitness. It adds to the ability of a virus to move through a population, to transmit, actually, because someone's walking around and they're healthy and they're shedding and they can infect you. So actually, if a virus became less virulent, if it made more asymptomatic infections, it could become more transmissible, right? That makes sense. Variants were popping up before vaccination occurred. Yes, they were around before vaccination. You can find them in the sequence database from early 2020 for sure. Thank you, Westfield, for your contribution to the incubator. And we've talked about the op-ed. It's out. I showed it before. I'll show it again here. There it is. Oops, I'm making it bigger. That's not good. There it is. There's the headline. Let me make that a little smaller here. Pandemic, human behavior during the pandemic is more important than any COVID variant. And there it is. So I can send it to you if you'd like. You send me your email address, vincent@microbe.tv, or Amy and I are going to publish, a version that contains more science, I think. I don't think I can publish the original. If I can, I will do that as well. Yeah, if you call to cancel at times, they will usually offer a lower rate. That's right. I know people who have canceled their Time subscriptions because they're unhappy with the COVID coverage. I don't think they've got it right most of the time, frankly, especially on the variants. But we've beaten to death, of course. We've allowed anti-vax to fester for decades, not doing anything about it. Well, people should try to do something about it, but the anti-vaxxers are well funded, and they know how to manipulate social media to get at the top of search engines and so forth. So it's not easy to combat them. Wakefield makes money doing this, makes a lot of money doing this. So, you people who recommend vaccination don't make money, so it's difficult. But I do think that we're making some inroads. But yes, it's a festering problem. Hirsch and Drosten think there's no such thing as population immunity. Well, why isn't there population immunity? Why wouldn't there be? There's population immunity for any infectious agent. I mean, even influenza, there's population immunity, and the virus changes and it avoids it. That's fine, but for a period of time, there is population immunity. So I don't know what he's talking about. He's a smart guy. He was on TWIV twice. I'm probably misunderstanding what he's saying. When do you think we'll have EUAs for younger than 12? The estimate is October-ish of this year in the U.S. Talking about subscription cancellations. What did I do? I had a Time subscription, and I don't know, went up in price. So I called to cancel it, and she said, and they tried very hard to talk me out of canceling it. They sent me to more and more people. Finally, I canceled it because I said I want to do it online myself with a PayPal account so I could cancel at any time. Because if you give them a credit card and you call, then they try and talk you out of canceling for a long time. It's a waste of time. You should have the right to cancel any subscription without hearing a harangue for 20 minutes. For God's sakes, I know that's their job to keep the money flowing, but really? Brett Weinstein, by the way, I don't know the subject here, but I was asked to debate him about the lab leak hypothesis. And I said, first of all, it's not a debate. And secondly, I'm not debating anyone but a virologist. So then they changed him for someone else who was also not a virologist. So I said, forget it. No, that's it. That's the thing Amy was asking about, the Federal Open Market Committee. Okay, I don't know. I don't know much about money. I don't think about money much. I want to give all my knowledge away for free because I think it should be accessible to everyone. So I don't think much about it. If 100% of the world's population receive vaccination, would there still be potential for variants? Yeah, because efficacy is not 100%. Infections will still occur. So yes, but you probably wouldn't have any disease, so that would be good. But then, the next year you couldn't get 100% again. So that's a theoretical question, I understand, but it's interesting. What are your thoughts about LA recommending masks for vaccinated and unvaccinated indoors? Well, if cases are going up, first of all, what is a case? What are they defining as a case? Just an infection, PCR positivity? I don't care about PCR positivity. Is it moderate to severe disease? Probably not in vaccinated people. The unvaccinated people are getting infected, and they should be wearing masks until they get vaccinated. Why aren't you vaccinated? Are you under 12? Okay, that's acceptable. They should be vaccinated. So I don't think if you're vaccinated, you need to wear a mask indoors. They're just freaking out about the Delta variant being more transmissible, and it's not. They're thinking, you better wear a mask. No. If you're vaccinated, that's good enough. Okay, we're getting to the Federal Reserve stuff. A lot of questions here. Great. Sorry that takes so long to get through them all. A lot of rumors that I don't want to deal with. And remember, if you want to ask us a question, you have to put our name in front because there's a lot of conversation. What happens when you run out of Greek letters? And they start putting numbers. But we have a ways to go before we run out, right? We're only at, lambda? I don't like the naming system. I think there's a better way. Okay, Amy doesn't like putting the name of the origin in it. Fine, don't put the origin in it. But there are better ways to do it than this way. I mean, this is fine for the public, but for scientists, not a good idea. R naught of delta is 2 to 3, just like the ancestral. If you want to recalculate it, all right. So here's the problem. There's the reproductive index R naught, which is the fundamental RO of the virus, and then you can reduce it by interventions. So you have to figure it out early on to try and get as close as possible to basic properties of the virus, and then later on it changes in an outbreak because of interventions and the number of people that get together. So now they recalculate the R naught for Delta, but they never look at human factor. They only looked at the number of cases. And I would argue that unmasking and failure to distance is going to increase the number of cases, and it's going to increase the R naught, but it's not a biological property of the virus. The virus is not more transmissible. It is being transmitted through the population because it's got increased fitness and because people are misbehaving. What are your thoughts on experts taking Pfizer besides taking J&J? There have been a few publications on J&J, AstraZeneca, Pfizer, Moderna, Pfizer, and that's okay. I don't see why it would be a problem to do both. I absolutely think it's fine. It's safe, and you're going to get a boost. It's not a problem. Thank you, Robbie, for your contribution. Really appreciate it. All goes to the incubator. What do we got? 9:15. Good. 15 minutes. Is there a foolproof approach to prevent any pandemic, or does an approach depend on the pathogen? So this, as I've said many times, this pandemic, COVID pandemic, could have been prevented from the early days. How? And we knew after SARS-1 that bats harbored SARS-like coronaviruses that were threats to people, right? So why not make an antiviral? You could find a target in the virus and make a drug that would hit a lot of coronas, including SARS-CoV-2. In fact, we have one, molnupiravir, that was developed before SARS-CoV-2 arose. It's now in people. It seems to be working well, orally delivered. Why the hell did we have to wait to now to develop that? Why couldn't that be developed before and stockpiled? Why? Well, companies not going to do it. No profit to be made. Governments won't fund it. Too risky. So we need to support that kind of research, and that's where CEPI and READY come in. CEPIs and READYs are nonprofit companies that fund. CEPI does vaccines, and READY does antivirals for targets that nobody else would touch, and develop them and get them to a phase 1 so that they would be ready. And you could quell the outbreak. Definitely. Can you imagine if we had a stockpile of molnupiravir at the beginning of this? We could have given many, many people the drug and stopped the spread. There's no question. That's the way to do it. Now, you could say, well, we could do surveillance, but we know there are coronas out there that are a threat, and they're going to emerge again. We know they're influenza viruses. We should be ready for that. So we won't be able to 100% prevent it. Well, you would prevent a pandemic. Let's put it that way. You would still have an epidemic, but not a pandemic, and that's pretty good, right? Yeah, it's probably safe and effective, but no studies supporting it, but I don't see why it would be an issue. Yeah, if you're here, you got 450, you just hit the like button. That would be nice. If you could do that, it would sort of transmit to the rest of YouTube that we're doing good stuff here. How likely will next year's flu season be bad due to relaxed masking? Well, I think we go back to the way it was before COVID, right? So some flu seasons are worse than others. It depends on the virus that's circulating, the variant that is circulating. Remember, every year a flu variant emerges and it displaces the previous one. Do the flu people say it's more transmissible? No. It's fitter, and that's why it's displacing. So I don't understand why there's this disconnect between flu and COVID. Anyway, I don't think it'll be worse because we're relaxing all this non-pharmaceutical interventions. Worried by the Delta variant, WHO officials urge vaccinated people to keep wearing masks. So WHO is also at fault for not understanding the data. You know, some epidemiologists in a country tell WHO, oh, it's 10 times more transmissible. WHO just believes it without looking at the data. The virus is more fit. It's simply displacing others. You don't need to wear masks if you're fully vaccinated under the right conditions. Oh, well. Let's see what else we have here. Scrolling down, looking for questions. That would be a broad interest. Don't be sorry if you didn't, then that's not a problem. It's just that I don't understand it, and I can't always answer it if I don't. Thank you, Wendy, for your contribution. Really appreciate it. I understand the WHO has saved a lot of lives, but they are not saving lives by saying variants are more transmissible, unless they think scaring people is a good thing. Do you think scaring people works? Does it get people vaccinated? You know, if all the anti-vaxxers got vaccinated because the WHO says the viruses are more transmissible, that would be cool, but I don't think that happens. I don't think that happens. Obviously a non-zero probability of a lab leak, but as time goes on, there's no evidence for it, so it really approaches zero very quickly. I don't see any evidence for it. I've looked at a lot of data, and there's nothing for it. Thank you very much for your contribution. I still like a thought about being clean in the bathrooms in public facilities, restaurants, etc. I don't know what you mean there, Mike. Being clean in the bathrooms. When you go to the bathroom, you should wash your hands. And if it's a public bathroom, boy, even more so. I could tell you stories about aerosols generated by flushing urinals and toilets. And, you know, all of us excrete viruses in our urine. But it's probably okay because we all have those viruses, right? I always carry a small umbrella with my backpack, yeah, for sure. And Amy carries a backpack, so she should have one. Thank you, Rob, for your contribution. It's really appreciated. Really appreciated. Thank you, Tom, for your support. Really appreciate it. You know, the idea that someone was working on a virus that became SARS-CoV-2 is not really tenable. You know why? I was the first person to make a virus from the DNA and to be able to manipulate it. So I've been doing that for 40 years. Yeah, you can't make SARS-CoV-2 from scratch. No way. Were you able to escape getting polio before the vaccine came out? So I was born in 1953. And when was my first vaccine? So I was probably vaccinated in 55, 56, 57, yeah. So yes, I didn't get polio, and then I got I got the oral vaccine in 1960, and I have my vaccine card for that. But I assume I got the IPV, but I don't have a card for that. What would more transmissibility look like? Very difficult to distinguish from, more fitness displacing other variants. Maybe, a true increase in the reproductive index when human behavior is factored in. So, if you're in a room with one person infected, you might be infected within minutes as opposed to 10 or 15 minutes. It's hard to know, really. Right now we're looking at fitness effects, though. That's a great question. Great question. Oh, thank you very much for your contribution. Good. I'm glad that's useful. Not always sure I'm useful, but I try. At least I try. You would frame and hang on your wall an unofficial course diploma. Yeah, I would do that with a TWIV logo or Microbe TV. Yeah, it's a Microbe TV course. Make a cool one there. ICURN here. Welcome. I'm curious to know the studies we have on how complicated or not the process is for these viruses to adapt to human hosts. Well, we don't have any studies because we don't do those kinds of things. We don't take an unknown virus and adapt it to humans, right? So here's the thing. Viruses mutate extensively all the time. So bats have a lot of viruses, and there are mutants all over the place. And just randomly, mutations can arise that allow those viruses to reproduce in humans. And we know that because we can see, for example, viruses from pangolins that have a spike that can bind human ACE2, and they've never been near a human or in a human. They haven't been selected in a human. So mutations occur randomly, and some of them allow the virus to infect people. And then what's needed is contact between the animal with that particular virus and people. And that's the part that's hard because many of these animals are isolated. They never contact people. So that's why it's rare. And in the case of SARS-CoV-2, somewhere there was a bat shedding almost SARS-CoV-2, and it infected a person, and maybe it spread a little bit more. So here's the scenario. A farmer goes into a cave and collects bat guano to fertilize their fields, and they inhale virus, and they get infected. Virus reproduces, and they go home. He infects his wife and kids. And then somebody goes into town, and they infect more people. So it's spreading person to person. Maybe you select for a virus that's now really good at transmitting person to person. That's the scenario that we're talking about here. But, it's based on what we understand about viruses and conjecture, because we cannot actually do those experiments. Thank you, Sandy, for your contributions. Really appreciate your support. My blue glasses look good. Yeah, I have different glasses here. I like to try them because they're reading glasses. So when I started this live stream, I had these, which are, the academic glasses. And then I decided to experiment, and I have those blue ones. But I bought these, and Amy says I should never wear them. These are the architect glasses. And, yeah, they don't fit me, but I think they're cool. So I wear them just by myself sometimes. There's a famous lady who wears black ones like this. Okay, so there you go. She's going to get mad at me for wearing those. And then I have these. I thought I would try them in orange and blue. I like these, though. They're cool. Pete Townsend, great guitarist. Oh, that's on the other side. All right, let's thank some people before we quit here. Lou Answine, thank you so much for your support. Really appreciate it. Can I stand in front of a green screen? Well, I could. It depends on the quality. I did some lectures in Leipzig a couple of years ago, recorded in front of a green screen. Oh, my God, they were amazing. They had the amazing setup. Looked great. I will do it only if it looks good. But I want to have a good environment at the incubator where the background is kind of like this, where I have actually this print on the wall here. Let me see if I can coordinate myself. That print is done by Michelle Banks, Artologica, and it's a watercolor of different viruses. It's blurry, right? Bokeh effect. I commissioned her to make a bigger one for the incubator, so I want to have that in the background. And I'm thinking of having a shelf with all my virus models, right, like these here. So I want to make a cool background. So maybe a green screen is not happening there, Okay, let's see what other. You guys have great questions, and I wish I could get through them all. It would take a couple of hours, and you don't want to stay here forever. But I know some people do 4-hour live streams. Thank you, Tom, for your support. And by the way, yeah, I did a TWEVO with Nels today, live streamed. And Nels is like hooked on live streaming. So that's cool. I got to get the TWIP team to do live streaming next. And tomorrow's TWIV dropping at midnight. You're going to like it. 2 special guests, and it's going to be a lot of fun. Pete, thank you for your support towards that standing table. Thank you very much. Yeah, I'm going to have a standing table and a sitting table. I don't want to move it up and down. I know that's cool, but I want to be able to move within. The studio is big enough that I could have one in one corner and one in another. Had expected transatlantic communication to be better. Drosten thinks R0 is 6 and the vaccine will not protect enough. He thinks it's 6. See? He doesn't know because the calculations aren't done properly. And he thinks it will not protect enough against transmission. I don't think that's correct. But we'll see. We'll see. I think protecting against disease is really, really important. Oh, listen, there's something here going on in San Francisco. Please join the Rose Society. Yeah, you can toast us. Cool. Sorry, I'll miss it. I'm looking for more supports to thank. I know there are a lot of great questions. Thank you, Post Polio News, for your. Thank you. I'm thinking of. I want to do what is it called? I can't even remember the name of it. All right, forget it. Cancel those previous stumblings. I can't remember what I wanted to try. Some podcasts have a club where you subscribe and then you get extra things like no ads in podcasts. Well, we don't have any to begin with. So I don't know what I would offer people. Perhaps your daughter could play at the start. Well, maybe Amy would let me play her music at the... before we start, we could have a counter running with the music. Sure. That would be a great idea. Thank you, Overjoyed. Really appreciate your support. Overjoyed watched Tweevil today, even though she was supposed to be working. Thank you, Miter1, for your support. Wow, folks, you guys are so generous. Thank you, Ralph, for your support. Powell, thank you so much for your support. Really appreciate it. And who else do we have here? I got to get everyone. There's so many great things I'd love to... all right. Yeah, we do have a Patreon. Microbe.tv/ No, patreon.com/microbe.tv. Thank you for deleting that one, Frank. I just noticed that one. Is a more transmissible variant ruled out, or there's no evidence? No, there's no evidence. And as I said, I've never seen a more transmissible virus ever emerge in an outbreak. It's possible, but I haven't seen it. It could happen here. But you need to provide evidence for this virus is biologically more transmissible. Epidemiologically, variants are moving through the population. There are outbreaks, but the outbreaks are caused by human activities. That's the whole point. I got an email from... Someone sent an email to my departmental administrator saying I should be fired for killing millions of people during this pandemic. Man, go figure. All right, folks. I say that because one of the comments was that I've done bad things, but I haven't, of course. Just trying to educate people. Thank you so much for joining us. Great questions. Thanks to the mods, Frank and Vanity Nutrition. And we'll see you next time. Be safe and have a good week.